Hello everyone, welcome to my channel and welcome again to another topic. So in this video, I will be discussing DNA replication. So at the end of this video, I am expecting you to learn something, and I hope that you learn something. So without any further ado, let's get to the discussion. In 1953, James Watson and Francis Crick introduced an elegant double helical model for the structure of deoxyribonucleic acid, or DNA. DNA, the substance of inheritance, is the most celebrated molecule of our time. Hereditary information is encoded in DNA and reproduced in all cells of the body. This DNA program directs the development of biochemical, anatomical, physiological, and, to some extent, behavioral traits. Early in the 20th century, the identification of the molecules of inheritance loomed as a major challenge to biologists. When T.H. Morgan's group showed that genes are located on chromosomes, the two components of chromosomes, DNA and protein, became candidates for the genetic material. The key factor on determining the genetic material was choosing appropriate experimental organisms. The role of DNA in heredity was first discovered by studying bacteria and the viruses that infect them. The discovery of the genetic role of DNA began with research by Frederick Griffith in 1928. Griffith worked with two strains of a bacterium, one pathogenic and one harmless. When he mixed heat-killed remains of pathogenic strain with living cells of the harmless strain, some living cells became pathogenic. He called this phenomenon transformation, now defined as a change in genotype and phenotype due to assimilation of foreign DNA. In 1944, Oswald Avery, Maclean McCarthy, and Colleen McLeod announced that the transforming substance was indeed DNA. Their conclusion was based on experimental evidence that only DNA worked in transforming harmless bacteria into pathogenic bacteria. Many biologists remained skeptical, mainly because little was known about DNA. More evidence for DNA as the genetic material came from studies of viruses that infect bacteria. Such viruses, called bacteriophages or phages, are widely used in molecular genetics in research. In 1952, Alfred Hershey and Martha Chase performed experiments showing that DNA is the genetic material of a phage known as T2. To determine this, they designed an experiment showing that only one of the two components of T2, DNA or protein, enters an E. coli cell during infection. They concluded that the injected DNA of the phage provides the genetic information. It was known that DNA is a polymer of nucleotides, each consisting of a nitrogenous base, a sugar, and a phosphate group. In 1950, Erwin Chargaff reported that DNA composition varies from one species to the next. This evidence of diversity made DNA a more credible candidate for the genetic material. Two findings became known as Chargaff's rules. 1. The base composition of DNA varies between species. 2. In any species, the number of A and T bases are equal and the number of G and C bases are equal too. The basis of these rules was not understood until the discovery of the double helix. After DNA was accepted as the genetic material, the challenge was to determine how its structure accounts for its role in heredity. Maurice Wilkins and Rosalind Franklin were using a technique called X-ray crystallography to study molecular structure. Franklin produced a picture of the DNA molecule using this technique. Franklin's X-ray crystallographic images of DNA enabled Watson to deduce that DNA was helical. The X-ray images also enabled Watson to deduce the width of the helix and the spacing of the nitrogenous bases. The pattern in the photo suggested that the DNA molecule was made up of two strands, forming a double helix. Watson and Crick built models of a double helix to conform to the X-rays and chemistry of DNA. Franklin had concluded that there were two outer sugar phosphate backbones, with the nitrogenous bases paired in the molecule's interior. Watson built a model in which the backbones were antiparallel. Their subunits run in opposite directions. At first, Watson and Crick thought that the basis is paired like with like, A with A and so on, but such pairings did not result in a uniform width. Instead, pairing a purine with a pyrimidine resulted in a uniform width consistent with the X-ray data. Watson and Crick reasoned that the pairing was more specific, dictated by the base structures. They determined that adenine is paired only with thymine and guanine is paired only with cytosine. The Watson-Crick model explains Chargaff's rules that in any organisms, the amount of A is equals to T and the amount of G is equals to C. The relationship between structure and function is manifest in the double helix. Watson and Crick noted that the specific base pairing suggested a possible copying mechanism for genetic material. Since the two strands of DNA are complementary, each strand acts as a template for building a new strand in replication. In DNA replication, the parent molecule unwinds and two new daughter strands are built based on the base pairing rules. Watson and Crick's semi-conservative model of replication predicts that when a double helix replicates, each daughter molecule will have one old strand, derived or conserved from the parent molecule, and one newly made strand. Competing models where the conservative model, the two parent strands rejoin, and the dispersive model, each strand is a mix of old and new. Experiments by Matthew Michelson and Franklin Stahl supported the semi-conservative model. They labeled the nucleotides of the old strands with a heavy isotope of nitrogen, while any new nucleotides were labeled with a lighter isotope. The first replication produced a bond of hybrid DNA, eliminating the conservative model. A second replication produced both light and hybrid DNA, eliminating the dispersive model and supporting the semi-conservative model. The copying of DNA is remarkable in its speed and accuracy. More than a dozen enzymes and other proteins participate in DNA replication. Replication begins at a particular site called origins of replication, where the two DNA strands are separated, opening up a replication bubble. A eukaryotic chromosome may have hundreds or even thousands of origins of replication. So replication proceeds in both directions from each origin until the entire molecule is copied. At the end of each replication bubble is a replication fork, a Y-shaped region where new DNA strands are elongating. So helicases are enzymes that untwists the double helix at the replication forks. The single-strand binding proteins bind to and stabilize single-stranded DNA. The topoisomerase corrects overwinding ahead of replication forks by breaking, swiveling, and rejoining DNA strands. The DNA polymerase cannot initiate synthesis of a polynucleotide. They can only add nucleotides to the 3' end. The initial nucleotide strand is a short RNA primer. An enzyme called primase can start an RNA chain from scratch and adds RNA nucleotides one at a time using the parental DNA as a template. The primer is short, 5 to 10 nucleotides long, and the 3' end serves as the starting point for the new DNA strands. Enzymes called DNA polymerases catalyze the elongation of a new DNA at the replication fork. Most DNA polymerases require a primer and a DNA template strand. The rate of elongation is about 500 nucleotides per second in bacteria and 50 per second in human cells. Each nucleotide that is added to a growing DNA strand is a nucleoside triphosphate. DATP supplies adenine to DNA and is similar to the ATP for energy metabolism. The difference is in their sugars. DATP has deoxyribose while ATP has ribose. As each monomer of DATP joins the DNA strand, it loses 2 phosphate groups as a molecule of pyrophosphate. The antiparallel structure of the double helix affects replication. DNA polymerases add nucleotides only to the free 3' end of a growing strand. Therefore, a new DNA strand can elongate only in the 5 to 3 direction. Along one template strand of DNA, the DNA polymerase synthesizes a leading strand continuously, moving toward the replication fork. To elongate the other new strand, called the lagging strand, DNA polymerase must work in the direction away from the replication fork. The lagging strand is synthesized as a series of segments called Okazaki fragments, which are joined together by DNA ligase. The proteins that participate in DNA replication form a large complex, a DNA replication machine. The DNA replication machine may be stationary during the replication process. Recent studies support a model in which DNA polymerase molecules reel in parental DNA and extrude newly made daughter DNA molecules. DNA polymerases proofread newly made DNA, replacing any incorrect nucleotides. In mismatch repair of DNA, repair enzymes correct errors in base pairing. DNA can be damaged by exposure to harmful chemical or physical agents such as cigarette smoke and X-rays. It can also undergo spontaneous changes. In nucleotide excision repair, a nuclease cuts out and replaces damaged stretches of DNA. Error rate after proofreading repair is low but not zero. Sequence changes may become permanent and can be passed on to the next generation. These changes, mutations, are the source of the genetic variation upon which natural selection operates. Limitations of DNA polymerase create problems for the linear DNA of eukaryotic chromosomes. The usual replication machinery provides no way to complete the 5 ends, so repeated rounds of the replication produce shorter DNA molecules with uneven ends. This is not a problem for prokaryotes, most of which have circular chromosomes, but in eukaryotic chromosomal DNA molecules, it has special nucleotide sequences at their ends called telomers, and telomers do not prevent the shortening of DNA molecules, but they do postpone the erosion of genes near the ends of DNA molecules. It has been proposed that the shortening of telomers is connected to aging. If chromosomes of germ cells became shorter in every cell cycle, essential genes would eventually be missing from the game test they produce. An enzyme called telomerase catalyzes the lengthening of telomeres in germ cells. The shortening of telomeres might protect cells from cancerous growth by limiting the number of cell divisions. There is evidence of telomerase activity in cancer cells, which may allow cancer cells to persist. The bacterial chromosome is a double-stranded circular DNA molecule associated with a small amount of protein. Eukaryotic chromosomes have linear DNA molecules associated with a large amount of protein. In a bacterium, the DNA is supercoiled and found in a region of the cell called the nucleoid. Chromatin, a complex of DNA and protein, is found in the nucleus of the eukaryotic cells. Chromosomes fit into the nucleus through an elaborate, multi-level system of packing. Chromatin undergoes changes in packing during the cell cycle. An interphase, some chromatin is organized into a 10 nanometer fiber, but much is compacted into a 30 nanometer fiber through folding and looping. Though interphase chromosomes are not highly condensed, they still occupy specific restricted regions in the nucleus. Most chromatin is loosely packed in the nucleus during interphase and condenses prior to mitosis. Loosely packed chromatin is called euchromatin. During interphase, a few regions of chromatin, centromeres and telomeres, are highly condensed into heterochromatin. Dense packing of the heterochromatin makes it difficult for the cell to express genetic information coded in these regions. Histones can undergo chemical modifications that result in changes in chromatin organization.
So, that's the end of the discussion. So, I hope that you learned something from the video, from the discussion that I have made, and see you on another video vlog or another lesson with me, Scout. So, that is all. Goodbye!